Cancer Center in Chennai, and Dr. Kajari Bhattacharya, who is the Associate Professor at Department of Radiology in Tata Memorial Hospital, Mumbai. The presenter for today's slide seminar is Dr. Vani Santosh, who is the retired professor at Department of Neuropathology from NIMHANS, Bangalore. These are our participants, the students who will be presenting the cases. Kindly introduce yourselves. A resident from Sims.
From Bardwan Medical.
Sir, Dr. Anita Borges, ma'am, and box tumors with emphasis on case-based studies.
Welcome. Good evening. Dr. Kajri is.
Yeah, okay. Are you all able to see this? See the screen?
Yes, ma'am.
Yes, ma'am.
Yes.
Okay, all right. Good evening to everyone. First of all, thank you, Sumit, for inviting me to. As interactive as possible today, and we are starting our session on brain tumors, and we'll begin with adult brain tumors and approach to adult brain tumors, but I will be emphasizing predominantly gliomas and a few other tumors like glioneuronal tumors. Now, before we actually start, let us look at the evolution of the CNS tumor classification itself. The very first classification was put forth by Bailey and Cushing. This was way back in 1926. A very primitive classification, nevertheless, it was quite a consistent classification, and therefore it lasted for a very long time, for nearly 50 years before the WHO took over the classification of the CNS tumors in the year 1979. And between these years, 1979 to 2007, the 4 editions of the WHO classifications have been published. These are popularly referred to as the Blue Books. And in all these, the classification was based solely on the histomorphology, and that was supplemented by immunohistochemistry in the later editions. But post 2007, what we have seen is, post 2007, what we have seen is that there has been an explosion of molecular data from which have evolved a growing number of molecular biomarkers with important diagnostic, prognostic, and predictive information. And some of these biomarkers were incorporated into the revised 4th edition, that is the WHO 2016 classification. And it was this classification that for the very first time included morphology and genetics in the classification for the first time. Earlier to this, all classifications had only histomorphology. And therefore, it brought about a paradigm shift in the classification of the CNS tumors, particularly some groups of tumors like diffuse gliomas as well as the embryonal tumors. Now, what we have seen post 2016 also is that there is a rapid advancements in neuro-oncology. And in order to keep in pace with it, the C-IMPACT now, which is the consortium to inform molecular and practical approaches to CNS tumor taxonomy, was announced in the year 2017. Now, this was by the WHO group itself, but it was not officially WHO at that time, and therefore now stands for not officially WHO. The goal of this consortium was basically to review the literature for new molecular markers, new biomarkers, new molecular alterations, and which have some clinical significance, and to decide which of these molecular markers can be incorporated into the subsequent classifications. There are 7 updates of the C-IMPACT now that have been published between the years 2018 and 2020, and all of them are published in Acta Neuropathologica, which is the official journal of the International Society of Neuropathology, and those of you who are interested can go through these updates. And it is the recommendations of all these updates that are now incorporated into the current classification that we are all following, that is the WHO 2021 classification. This is the 5th edition and very popularly referred to as the WHO CNS5. And therefore, this classification advances the role of molecular diagnostics even further in CNS tumor classification. Nevertheless, it is deeply rooted to established approaches of tumor characterization. So what are those established approaches? it is histology and immunohistochemistry. So we still have histology and immunohistochemistry, which plays a very important role in the classification even in this 5th edition, and therefore the classification presents a hybrid taxonomy to us. So let us look at some of the major changes that have happened in this WHO CNS 5. So some of the major changes include revision in CNS tumor taxonomy, CNS tumor nomenclature. We'll discuss each one of these. There is revision in the CNS tumor grading and introduction of new tumor types and subtypes. Every time a new classification comes out, there will be some new tumor types and subtypes that gets introduced. And there is the use of the two terminologies not otherwise specified, NOS, and not elsewhere classified, that is NEC. And the use of a very new high throughput molecular technique known as DNA methylation profiling that is used for the diagnosis of some of the tumors, and the use of what is known as layered reporting or the integrated diagnosis format. So all these are the major changes that have happened. Let us look at each one of them very briefly. First is the revision in CNS tumor taxonomy. Now the reason this classification is hybrid is because some of the tumors are diagnosed based on their underlying genetic alterations, that is, their genetically defined tumor types. For example, astrocytoma IDH mutant, diffuse midline glioma H3K27M mutant, or astroblastoma MN1 altered. All these are defined by their genetic alterations. So you cannot make a diagnosis of these tumor types without demonstrating these genetic alterations. But there are some tumors where we can diagnose them only based on histopathological features like meningiomas or pilocytic astrocytomas or ganglioglioma. Sure enough, they do have genetic alterations, but that is not required for the diagnosis of these tumor types. And there are some tumor types that can be diagnosed based on both histological features and molecular features. For example, medulloblastoma. We have two groups in medulloblastoma, a histologically defined group which has all these tumor subgroups such as classic, the desmoplastic nodular, the large cell anaplastic, and we have this molecularly defined medulloblastoma which has the subtypes such as the Wnt activated, SHH activated, and others. So these are diagnosed based both on histological as well as the molecular parameters, and that is the reason this classification is a hybrid classification. And in order to standardize this WHO CNS 5 with the other 5th edition blue books, some terminologies have been changed. For example, we don't use the term entity anymore. In the earlier classification, they were all referred to as tumor entities, but now we call them as tumor types. And histological variance is now designated as subtypes. So we have only type and subtype. We do not have any tumor entities and tumor variants, and it's the types that are listed in the classification. Now there is revision in the CNS tumor nomenclature based on certain modifying factors. If you look at anatomic site modifiers, for example, there is this tumor called chordoid glioma. In the earlier classification, this was chordoid glioma of the third ventricle. But the term third ventricle is now deleted because now we know that chordoid gliomas can occur anywhere else also in the brain, not just in the third ventricle. And there are some nomenclatures have altered based on genetic modifiers. For example, glioblastoma. Now earlier we had glioblastoma IDH wild type and we had the glioblastoma IDH mutant. But IDH wild type is retained whereas the mutant is removed. Supratentorial ependymoma rela fusion positive is retained. Now it is known as Zefta fusion positive because there is a change in the name for the genetic alteration. And there is this embryonal tumor known as ETMR. It is embryonal tumor with multi-layered rosettes. It is characterized by a particular genetic alteration known as C19MC alteration. So in the earlier classification, this tumor was designated as ETMR C19MC altered. But now the C19MC alteration is removed because now we know that ETMRs can have other mutations also, like DICER1 alterations. Other mutations are known in ETMR. So we call it just ETMR. So there are changes in this or revisions in the CNS tumor nomenclature. And WHO CNS 5, it pays a lot of importance to gene and protein nomenclature and uses this Hugo Gene Nomenclature Committee and Human Genome Variation Society recommendations for gene symbols, names, and sequence variants. You should remember this. Gene symbols are all presented in italics, but the proteins and the gene groups are not italicized. And we prefix this letter C for any gene sequence alteration and the letter P for protein sequence. Let us look at one example. The most common genetic alteration in adult diffuse glioma is IDH1R132H mutation. What is the gene sequence alteration? Gene sequence alteration is IDH1C dot 395G2A. That means there is a transition from G2A in nucleotide position 395, and that results in this protein sequence where you have IDH1P dot R132H, where arginine is replaced by histidine in position 132 of the amino acid sequence. Now there is revision in CNS tumor grading in this WHO CNS file, and we now use Arabic numerals for grading and not Roman numerals. The reason Arabic numerals are used like this is because it has to be made uniform with the other WHO grading systems. In all other WHO grading systems, it's Arabic numerals. So even in CNS, it has become Arabic. And moreover, the Roman numerals used to give rise to some transcription errors. For example, for 1 could be typed as 2 or 2 as 3, some sort of transcription errors. And the term CNS WHO grade is introduced for every tumor type. So if you want to grade it, you have to use the CNS WHO grade. So for example, ependymoma. Ependymoma is a CNS WHO grade 2 tumor. So like that, you have to use this terminology known as CNS WHO grade. And there is grading within the tumor types. For example, astrocytoma IDH mutant is graded as CNS WHO grade 2, grade 3, grade 4, and that replaces the previous terminologies. Diffuse astrocytoma was what we used to, what was designated for this grade 2 tumor, anaplastic for grade 3, and IDH mutant glioblastoma for grade 4. So we don't use these terminologies anymore. And the term anaplastic is eliminated for grade 3 tumors. For example, pleomorphic xanthoastrocytoma, it has 2 grades, grade 2 and grade 3. Grade 3 used to be designated as anaplastic PXA earlier, but now we don't call it anaplastic PXA. You just call it PXA CNS WHO grade 3. A combined histological and molecular grading is applied to some tumor types. For example, astrocytoma IDH mutant. We'll come to these tumors. Now, we know that we can grade these tumors based on histological parameters, and we look at some of those histological parameters. But now we have one molecular parameter also that is included for grading, that is CDKN2AB homozygous deletion, and we'll look at this a little more in detail subsequently. Glioblastoma IDH wild type. For grading, we know the histological parameters, but apart from histological parameters, there are these molecular parameters such as EGFR amplification, third promoter mutation, gain of chromosome 7, loss of chromosome 10, copy number changes that that will be used along with histological parameters for this glioblastoma IDH wild type. And WHO does not assign grades if it confuses clinical care. For example, diffuse leptomeningeal glioneuronal tumor and astroblastoma MN1 Lauter, they are not assigned any grade by WHO because even now, till now, we do not know exactly how these tumors behave, whether there are subsets of the tumors that behave in a better manner, subsets that do not behave in a better manner. Therefore, a grade is not assigned to these tumors. So as I told you, in every classification, there are new tumor types and subtypes will be introduced. And in this classification, we have 22 newly recognized tumor types. 14 of them come under the group of gliomas and glioneuronal tumors, and 8 under the other groups. And let me tell you that the majority of the newly recognized tumor types are all in the pediatric age group, as you can see in these boxes. Now, we also use this terminology NOS and NEC, which is allowed by WHO. So what is NOS? NOS is not otherwise specified, which implies that the necessary diagnostic molecular tests, they were not available, they're not performed, or the results are inconclusive. That is, you are in a center where you couldn't do this molecular test, and so you can use this terminology NOS, and that's only a red flag. It will prompt additional testing, that this case has to be tested for a particular molecular alteration in another center. NEC is not elsewhere classified, where you did all the necessary molecular tests, but the results, they do not allow for a proper WHO diagnosis. That is, there is some kind of a histologic or genetic mismatch. In both these situations, one can consider what is known as a layered reporting format. This is very interesting. Have a look at the layered reporting format. A layered reporting format is one that optimally combines morphologic and molecular data to generate a layered neuropathological diagnosis. And this is the example of the template for a layered diagnosis that is put forth by WHO itself, which has 4 layers, as you can see here. The first layer is the integrated diagnosis. This is typically a WHO diagnosis. It has, whether it is genetically defined, histologically defined, it should be a WHO diagnosis. If you cannot reach this diagnosis, then you call it as NOS or NEC. And if you, the second layer is histopathological classification that is based on histology, immunohistochemistry. Mind you that this can be different from the integrated diagnosis. That is, for example, you have a tumor in a pediatric age group where the histopathology looks like oligodendroglioma, but the molecular alterations are totally different. It is not at all an oligodendroglioma. It could be a MAP kinase altered pediatric type of low-grade glioma. So this is a histopathological classification. And CNS WHO grade is an integrated CNS WHO grade. That is, you should have this integrated WHO diagnosis to call it CNS WHO grade. If you have an NOS diagnosis, then you cannot call it CNS WHO grade. You have to say histological grade. And the last layer is molecular information, where all the molecular findings are listed along with the genetic platform that you use for the molecular testing, like the whether you used immunohistochemistry for the surrogate markers, genetic assays like FISH or the Sanger sequencing, next-gen sequencing, methylome profiling, whichever assay you use, you can list that assay. Let us look at 2 examples. This is the first example. This is the integrated diagnosis is a complete WHO diagnosis here. It's oligodendroglioma, IDH mutant and 1p19q co-deleted. Histopathological classification is oligodendroglioma and the grade is 2. I've just taken an example of a grade 2. And the molecular information here is that the tumor is positive for IDH1R132H, which is a surrogate for IDH mutation. So we know that there is IDH1 mutation. And 1p19q is co-deleted by the FISH technique. So because we have all the molecular information, we are able to give an integrated diagnosis and we are able to give a integrated CNS WHO grade. There is another example which same oligodendroglioma, which is IDH mutant, but I've called it NOS. Now why have I called it NOS? Because here we have information that it is IDH1R132H positive by IHC. In my center, I was able to do only IHC. And so it is, we know it is IDH mutant, we know it is oligodendroglioma by histology, but I am not able to perform FISH on this particular case in my center. So since it's not a complete diagnosis, we have to say NOS. And once you give a NOS, you cannot give a CNS WHO grade. You have to give a histological grade. Because CNS WHO grade is given only when you give a complete integrated diagnosis. I hope this is quite clear to the students. Now, what WHO CNS 5 has done is it has introduced this essential and desirable diagnostic criteria for every tumor type. Essential diagnostic criteria are those that are definitive criteria. That means they are must-have feature for the diagnosis of a particular tumor. For example, for astrocytoma IDH mutant, IDH mutation is an essential diagnostic criteria. Whereas desirable diagnostic criteria are those that can support a diagnosis, but they are not mandatory for the diagnosis. For example, for this tumor called pleomorphic xanthoastrocytoma, large majority of them will show this BRAF V600E mutation. Okay, it is characterized by BRAF V600E mutation. But let me tell you, it is not seen in 100% of the cases. It's seen in about 70 to 80% of the cases. And that is why this is a desirable criteria. You can still make the diagnosis of PXA on histology without BRAF V600E mutation demonstration. So this becomes a desirable diagnostic. So every tumor will have a essential criteria and a desirable criteria. Okay. And now, what is this DNA methylation profiling? This is a very high throughput methylation profiling technique. What WHO CNS 5 has done is it has used this technique to develop the classification. Now, these DNA methylation profiling experiments were carried out way back in the year 2018 by the German group, by the Heidelberg group, where they profiled more than 2,500 tumors, okay, and they generated a huge data because it's high throughput. You use a particular platform which will profile the entire methylation sequence of that particular gene. And what they found was they had a huge data that was generated, and they used appropriate bioinformatic tools, and they were able to plot this atlas where tumors could be classified very beautifully. And they showed that the methylation based classification was much superior to the histology based classification. But we know that we can't use this for routine diagnosis. The WHO CNS5 doesn't recommend that we use it for every case for routine diagnosis. Sure enough, it is a very effective way to characterize some of the very challenging neoplasms and to identify some new or rare tumor types and subtypes. But we know that it is technically challenging. You and me cannot perform in all the centers. In fact, there are so many centers which have very basic IHCs. They don't even have surrogate IHC markers. So where can we perform DNA methylation profiling? And of course, there is the cost factor also. So what WHO CNS5 has done is, it has made DNA methylation profiling as a desirable criteria for most of the tumors. It's not essential. It is essential for very few tumor types. I've given just two examples. One is high-grade astrocytoma with pyloid features, and one is a new tumor type known as the diffuse glioneuronal tumor with oligodendroglioma-like features and nuclear clusters. You don't have to remember this. Just remember that it is an essential criteria only for few tumors and a desirable criteria. We still can make the diagnosis without this DNA methylation profiling. Now you can see this is how the tumors are classified in WHO CNS5. We have these 12 major groups of tumors. Each major group will have these smaller groups of tumors, and each smaller group will have tumor types, several tumor types. So I am supposed to be discussing the adult type of tumors, particularly with reference to these gliomas. But let me tell you that tumors that occur in the adults can also occur in the pediatric age group and vice versa. Nevertheless, now what are all in the yellow fonts are the tumors that predominantly occur in the adults. Adult type diffuse gliomas, cranial and paraspinal nerve tumors, meningiomas, mesenchymal non-meningothelial neoplasms, melanocytic neoplasms, hematolymphoid tumors, tumors of the cellular region, and metastatic tumors. These are predominantly in the adults. And what are in the white fonts, that is the pediatric type, low and high grade gliomas, ependymal tumors, embryonal tumors, and others, they're predominantly in the pediatric age group. And the ones that are in the orange fonts, like the circumscribed astrocytic gliomas or the glioneuronal and neuronal tumors, they occur in the young adults or sometimes in the older children. And as I told you, each of these tumor groups can have several tumor types. For example, the adult type diffuse glioma has 3 tumor types, which we are going to discuss. The group of glioneuronal tumor has 14 tumor types. So every group will have several tumor types. Some of them are genetically defined, some of them are histologically defined, and some of them are both genetically and histologically defined. So now we will focus on gliomas, because that is what we are going to look at, and mainly the adult type of gliomas. And this table, it shows you how the gliomas were classified in WHO 2016 versus this WHO 2021, which is WHO CNS5. In the earlier classification, all the diffuse gliomas, they came under this header called as diffuse astrocytic and oligodendroglial tumors. It had several molecularly defined tumor types. Of course, NOS was also used at that point of time. Almost 15 molecularly defined. They were called as tumor entities at that time. Now we call it as tumor types. And all the other circumscribed tumors came under this other astrocytic tumor. So this was a previous classification. But now you can see that the classification is much simplified. We have these groups: adult type diffuse glioma, pediatric type diffuse high-grade gliomas, diffuse low-grade gliomas, and circumscribed astrocytic gliomas. And each of them will have several tumor types within that group. So one of the major changes in this glioma classification in WHO CNS5 is the division of these diffuse gliomas into adult type and pediatric type. It was not there in the previous classification. Only now we have adult type and pediatric type, because we know that they're molecularly distinctly different neoplasms. And the division of pediatric diffuse gliomas into pediatric type high-grade gliomas and pediatric type low-grade gliomas. However, you should remember that the pediatric type diffuse gliomas can also occur in the adults, and the adult type diffuse gliomas can occur in the pediatric age group, although their genetic background is mutually exclusive. For example, adult type diffuse gliomas are heralded by these mutations, either IDH mutation or 1p19 and or 1p19q co-deletion, whereas the pediatric type diffuse gliomas, they usually harbor histone H3 mutations, or they may show alterations in the RAS MAP kinase pathways. This is the most common alteration that you see in this lymphopenia. Of course, there are so many other alterations also that have been described. So please remember that the tumors that occur in pediatric age group can occur in the adults. Adult age group can also occur in the pediatric age group. So now let us come to our group of tumors, that is the adult type of diffuse gliomas. And let me tell you that this is a genetically defined tumor group. The 3 tumors are genetically defined. Which are the 3 tumor types? It's astrocytoma IDH mutant, oligodendroglioma IDH mutant and 1p19q co-deleted, and glioblastoma, that is IDH wild type. Now, astrocytoma IDH mutant has 3 grades. I told you there is grading within the tumor type. The 3 grades are referred to as CNS WHO grade 2, grade 3 and grade 4. And what was called as diffuse astrocytoma IDH mutant grade 2, anaplastic astrocytoma and glioblastoma are no longer used. So you don't use these terminologies anymore. You don't use Roman numerals for grading. You use only Arabic numerals for grading. So this is what we are following, and this has become obsolete. And astrocytoma IDH wild type, this is eliminated. So we don't have any astrocytoma IDH wild type in the adults. These become glioblastomas, and if it is or can be any other pediatric type of an astrocytoma IDH wild type tumor, which I am not going to discuss. But remember that astrocytoma IDH wild type terminology is eliminated. What about oligodendrogliomas? Oligodendrogliomas are defined by IDH mutation 1p19q co-deletion. You cannot make a diagnosis otherwise. And they have 2 grades, CNS WHO grade 2 and grade 3. And we don't use the previous terminologies and the previous grading. Anaplastic oligodendroglioma, the terminology is not used. You just say oligodendroglioma, IDH mutant 1p19q co-deleted, CNS WHO grade 3, and don't use the term anaplastic oligo. Okay. What about glioblastoma? Glioblastoma is only IDH wild type. Earlier we had IDH wild type and IDH mutant. That is not there anymore. Now it is only IDH wild type, and you designate it as CNS WHO grade 4, and 4 is denoted in the Arabic numerals. I hope this is clear to you. The 3 genetically defined tumor types in adult type of diffuse gliomas. Okay. So with this we will, I think with this introduction, if you have any doubts at this point of time, maybe you can put it in the chat box, or we can discuss it subsequently. But we'll start with the cases, because then all these will become more and more clear to you. So the first case, case number 1 will be discussed by Siddhi. Siddhi, are you there?
Yes, ma'am.
So you can discuss this case.
Okay, so we have 32-year-old man presented with generalized tonic-clonic seizures for 2 months, headache and vomiting for 15 days, and left-sided weakness for 15 days.
What can you make out of this history, Siddhi? So yes.
The tumor is basically belong to the adult age group.
Yes.
And it is presenting with seizures, so it might presses any brain organs at there. So radiologically we can see something.
No, don't worry about radiology. Let us look at only the history. So there is a generalized tonic-clonic seizures in this patient, isn't it? So maybe white and gray matter of the brain is involved. And look at the duration of the symptoms. What do you think?
It is on acute onset.
Acute onset, very good. So it is not a very chronic one, isn't it? It's not going on for many years. It's acute onset. And then the patient has headache and vomiting for 15 days. What do you think that is because of?
That is because of the lesion that presses and now or
no, because of the increased intracranial pressure. There will be increased intracranial pressure, and that is why there is headache and vomiting, and there is a left-sided weakness, which means that the lesion is located on the
now
on the right side, isn't it? On the right side, it's located on the right side. Anyone else also can answer. I have no problem with that. Okay, so that is what the history tells us. Let us look at this image, and can I request Kajri, can you just briefly describe this image to them?
Sure, ma'am. So can I annotate actually?
Yeah, please.
So request.
Do you want me to annotate? Yeah, let me just pointer option. I'll take the pen. Okay.
So the thing is that there are 4 sets of actual images. So what we are seeing basically, first is written as a T2 flare, and the third is written as T2. So whenever we are commenting on any kind of brain MRI imaging,
Kajri,
yes, sir?
you can request for annotation from
Yes, yes,
Dr. Vani.
yes, yes.
Yeah, I will annotate. As she's telling, I'll annotate.
No,
Yes, Kajal.
ma'am, I have got that annotation, whatever that controls. So I'll start now.
Yeah, sure.
T2 weighted image. So it is very important for all the residents to know that the base image for any kind of MRI brain is a T2 weighted image. T2 weighted image gives you an idea about where is the location of the tumor, whether it is located in the gray matter, in which side it is located, and what is the tentative histology that we are expecting. Because what happens is that generally lower grade tumors, they are more homogeneous. That means they have a similar signal on, so they are generally homogeneous and there is generally a little bit hyper intense on T2. As the tumor grows high in grade, it has some intrinsic hypo-intensity. And when I'll show my cases, I'll show you how do the tumors evolve from lower grade to higher grade. So basically coming to this case, what we see is there is a lesion here which is present in the right insula region. And it is also, so this is the region of the basal ganglia and this is the insula if we compare on the normal side. On the contralateral side, so this is another thing, this is the left and this is the right when we are looking at MRI. So here we are looking at the right region which is involving the insular cortex. It is also involving the basal ganglia and the posterior limb of internal capsule. So all these clinical features is explained. Number one, patient has generalized tonic-clonic seizures. So as we can see, the cortex is involved, so therefore that can lead to your generalized tonic-clonic seizures. We can see that there is involvement of the posterior limb of the internal capsule, which is this on the left side. So that is involved, and therefore the patient is having weakness on the contralateral side. And other than that, we can see there is some midline shift. So we can see where this tumor is exerting some mass effects and it is causing the midline shift to the opposite side. So therefore the patient has features of raised intracranial tension, and therefore patient is having vomiting and headache. So this is the first thing. Other than that, we see that the tumor is basically hyperintense, or it is bright, or it is giving a white signal of T2 weighted energy. Next, we see this flare. So what we see is this is your T2 in which the lesion predominantly appears bright. In flare, there are some areas of inversion. Inversion means that whatever was appearing bright on T2 is showing some amount of signal drop. It has become a little bit hypo-intense on flare. What it signifies is that basically, if you see here, these two sequences are exactly similar except the CSF signal. So if you see that the fluid is hyperintense on T2 and it is black in flare. So whatever happens is that means that the fluid content of this tumor is a little high, and it is very often seen in, it's a very specific sign which is called as the T2 flare mismatch, which is seen in IDH mutant astrocytoma. Also, there is some grade of it. So if it's more than 50%, it is considered 100% specific of IDH mutant astrocytoma. So the sensitivity of this sign is low, it is around 40%. So apart from that, other sequences that include diffusion and SWI, but we don't have them. So what we see is a T1 weighted image. This is needed when we see the post contrast. So what we see is basically this tumor is hypo-intense on T1. So it's dark. So hypo means dark. So it's hypo-intense in T1. And what we have to do is we have to compare it with this side, that is the post contrast. We see there are series of post contrast enhancement. Enhancement means these are appearing a little bit brighter. And if we just corroborate it with this particular image, these are the areas which are a little bit dark. So there are some areas which are showing some features of higher grade, that is, they are appearing a little bit dark on T2 and they are showing some enhancement. So that's like, so another very important thing is when you look at the margins of this tumor, so I would, how would you remove the annotations and rub this? Is there any way? Okay.
I'm not sure, but
Okay.
Yeah.
Margins of the tumors, if you see, they are not very clear. They are not crystal clear. And when we come to later cases, we will see that these the margin of the tumor is very well seen. So this is a very the margin of the tumor is not that crystal clear, and that is what therefore it's a diffuse. So basically, this is a diffuse astrocytoma, and due to this mismatch, I would say that it is a IDH mutant astrocytoma. There are some areas of higher grade, is what I see. Now I request Pani ma'am.
Yeah, so she has described very beautifully. I hope you were able to understand, all of you. So she has shown you what these images are, and she's told you it is hyperintense on this T2, and whatever was hyperintense had to be hyperintense here also, but there is an inversion, and that is known as T2 flare mismatch, and one of the features of IDH mutant astrocytomas on radiology. So that is actually, it is a radiological biomarker. So let us go to the next one.
Just click in the center. Just click in the center of the screen. You'll next slide. Click in the center. Just click in the center of the screen.
Okay.
Next slide.
Okay, that's good. That's good. So now, Siddhi, can you describe? We have some histology images. So the lesion was subtotally resected, and then you can see that this is how the lesion is. This is how the histology is. Please describe.
So we can see here as various size cells which might be hypercellular areas, and somewhat mitosis is also there, and abundant eosinophilic cytoplasmic cells are also seen, and some areas showing vascular proliferation also.
So what do you think these cells are?
These cells are astrocyte cells.
So you should say that first to begin with only, isn't it? So if you just say cells, no one can make out. See, whenever you describe histology in any tumor, whichever, first you have to talk about the tissue pattern, cell type, and cell characteristics. Okay. So this is sort of a cellular tumor. You can see here it is very cellular on this right one, isn't it? So it's a, let me just change my pointer option. I'll take a laser pointer now. So here it is very cellular. It is infiltrative, and then you have a stroma in the background, isn't it? Thin fibrils are there in the stroma. These are all, what stroma do you think that is? What is that stroma? Is it a mesenchymal stroma? Is it a glial stroma?
Glial
stroma. It's a glial stroma, isn't it? So they're all infiltrating on this glial stroma, which are having these small spaces here. Can you see the spaces in the stroma? What are these spaces? They are referred to as microcystic spaces. So you have a fibrillated glial microcystic stroma over which these astrocytic cells. You can see that different cell types you can see, but all of them have an astrocytic morphology. This is very cellular, and then you can see that some of them, there is nuclear atypia, very significant nuclear atypia. What do we mean by nuclear atypia? Increase in the size of the nucleus, multinucleation, hyperchromatic nucleus, intranuclear cytoplasmic inclusion, all that comes under nuclear atypia. So there is significant, you can see some of them have abundant cytoplasm like a gemistocyte, some of them are fibrillated, some of them are roundish, almost look like oligo, isn't it? See here? Some of them are looking like oligodendrocytes also. And you're right that there is mitotic activity over here. Okay. So that is good. We'll go to the next slide. Why this is not moving. Let me change the pointer option and
okay. So go to the laser pointer.
No, I was in laser pointer, but
just click in the center. Click in the center of the screen, and next move now. It is not moving.
No.
Or maybe you'll have to erase everything what you have written. Just click on the right side. And just go to the pointer option, just say erase, eraser, all ink on the slides now, all ink.
Yeah, I'll erase all ink on the slide. Okay.
I think so.
Now I'll go to pointer option, and I'll just take a laser pointer now. Let us see whether it comes. No, it's not coming. So I'll erase this.
Oh, very good.
Maybe I'll just take an arrow option, which is automatic.
What happens when you click in the center? Can you move? You can't move the next slide?
No, it's
Can
not.
you stop sharing and share again?
Okay.
Yeah, you can share again now.
Sorry about that. Okay, I'll share this again.
Perfectly fine.
I'll share again.
Yeah. After a long time, we have good audience today. So 273 students to listen to you.
Sumi, there is some problem with this.
No. It will just click in the center of the screen, and move your from the key from the keyboard. Next.
Okay. Okay, I got it. There you go. Okay. Now this is a higher magnification. Siddhi, can you describe the cell types over here?
Yes, there are hypercellular areas which contain astrocyte with highly atypical cells, which some of them have abundant eosinophilic cytoplasm, some are showing mitotic activities, some are showing bionucleation and vascular proliferation also seen.
Where is vascular proliferation, Amma? There is no vascular proliferation. There is no blood vessel. I am not able to see.
Sorry, ma'am.
There are microcystic spaces.
Okay, ma'am.
Okay, these are thin vessels. They are the native vessels. They are proliferating. The thin vessels are there, but you are not seeing any microvascular proliferation. I'll tell you what is microvascular. Have you seen this multinucleated pleomorphic cell?
Yes, ma'am.
Yeah, so binucleate, multinucleate, astrocytes, gamestocytes, and brisk mitosis. In one field, you can see two atypical mitosis, isn't it? So let us look at the next slide now. This is again the same thing. You can see this abnormal mitosis. So many mitotic figures over here. It's very malignant. And you can see this pleomorphism. You can see binucleate cells. Some have abundant cytoplasm and some have this. So this is the higher magnification. So I've given you some diagnostic IHC markers. I've given you these two markers. Siddhi, can you tell me what are these and what does it show?
The GFP IHC is positive for.
What is GFAP? What is GFAP?
Glial fibrillary active protein.
Glial fibrillary acidic protein. Very good. GFAP is glial fibrillary acidic protein. Write down. Okay. Okay, it's an intermediate filament protein. And what has happened here? What does it show in this slide? In this,
Positive for the malignant cells.
no. Come on, tell me properly. Only cells are positive. Or only anything else is positive?
Background is also positive.
Very good, very good. You can see that the cells cytoplasm, the tumor cell cytoplasm is positive, plus the fibrils that come out of those cytoplasm, which form that fibrillated stroma. The whole thing is positive. Okay, that is what GFAP will stain. It will stain not only the cytoplasm, but all the glial fibrils. And that is why it's a glial tumor. It is GFAP strongly positive. That is why we say it is GFAP positive. And what is this? Olic 2, and you may not have seen this. I will explain. This is Olic 2. You can see a nuclear stain which is diffusely positive. Olic 2 is a very good stain that we use for all these diffuse gliomas. You can ask me what is Olic 2. Olic 2 is a very important transcription factor. It is essential for the specification and differentiation of these cells for oligodendrocytes, astrocytes, and neurons during development. So Olic 2, it gets universally expressed in all the differentiated oligodendrocytes, in astrocytes, in oligodendrocyte precursor cells, or what we call as oligodendrocyte-like cells. It will be positive in them. In immature neuronal and neurocytic cells are also Olic 2 positive, but it is not expressed in differentiated neurons, primitive neuroectodermal cells, ependymal cells, or microglial cells. So Olic 2 is negative in all that. So Olic 2 is always positive in these diffuse gliomas. So even in some of in the circumscribed gliomas also it will be positive. So for example, you want to differentiate a pilocytic astrocytoma from an ependymoma. Okay. And ependymoma will be Olic 2 negative, whereas pilocytic astrocytoma will be Olic 2 positive. So it's a very useful stain, and we use it for all these diffuse gliomas. So that is why I showed you that it is you use GFAP and Olic 2. And what is this stain, Siddhi?
This is MAP1, also called as KI67, which is used for counting the proliferating cells, counting the mitosis.
Not mitosis. Mitosis is different, proliferating cells are different. Okay, because when a cell proliferates, it goes from G0, G1, G2, S phase and M phase. M phase is the mitosis. But cell in proliferation can be in any of those phases. And this antibody will label those cells which are proliferating. It may not be in a mitotic phase, but proliferating. Okay. Understood? So, yeah, and MIB1 is actually a commercial antibody. It is from a company called DACO against the Ki67 antigen. You can use any other antibody also. So, Ki67 antigen will label the proliferating cells. It does not label the mitotically active cells. It will label them also because it is labeling all the proliferation. If you want only for mitosis, there is a histone marker called as PHH3. Okay, that is known as a mitotic marker, but not Ki60. It's not a mitotic marker. So the way we make a labeling index is we'll count about 1,000 cells, particularly near the hot spots, and then we make a percentage. And 1,000 cells, how many are positive? Make a percentage of that, and then you'll get your percentage. And in this case, it was more than about 12 or 14%. It was very, very high in this particular case. So what is your preliminary diagnosis and how do you want to proceed to arrive at the final diagnosis? What is your preliminary diagnosis?
WHO astrocytoma, WHO grade 3, IDH mutant.
Where am I? I didn't even show you IDH. How did you call it IDH mutant? Because Kajari told you. I didn't show you at all. Kajari, you gave away the diagnosis. You cannot. I have not shown you. I have just shown you the diagnostic markers. It is astrocytoma.
Grade 3.
That's all. You cannot say IDH mutant. So you want to proceed with the case, isn't it? So
Yes.
the preliminary diagnosis here will be a diffuse glioma compatible with astrocytoma. All right. NOS. Now we use NOS because we don't have any molecular markers and it's a molecularly defined tumor. Okay, we don't have a molecular marker, so we call it NOS. And because we are calling it NOS, we are giving a histological grade. And histological grade is 3. Did you understand? Students, have you understood?
Yes, ma'am.
So now let me ask you one more question. What next? What are we going to do? We are going to use surrogate IHC markers. So do you know of any surrogate IHC markers? You gave away the diagnosis initially.
We can use IDH.
Very good.
Then T53 we can use for.
Very good. Very good. And then
Then we can use ATRX.
very good. Excellent. 100 marks for you.
Thank
For
you.
this particular question, you've got full marks.
Thank you, ma'am.
So that this is the first slide. First one, whatever you said. What is this?
Vani, sorry to interrupt.
Yeah.
There was a very interesting question. It actually crossed my mind also. Why not
Yes.
grade 4?
We'll come to that. We'll come to that.
CDK in the late.
Yeah. We are going to come
At
to
least, at least I'm happy one of the participants has thought about that.
Very
Because
good.
of the mitosis, MIB1
So-
index.
Yeah. So let me tell you that if you're going to do a histological diagnosis and if you wanted to call it grade 4, you had to see one or two more histological features. That is necrosis and or microvascular process. Then we could have called it grade 4 on histology itself. But now on histology, we are not calling it grade 4 because if we go back, we haven't seen that. No, we haven't seen microvascular proliferation and we have not seen necrosis. So histologically, it's not a grade 4. Histologically, it's a grade 3 tumor. But then we know that this is a molecularly defined tumor type. So that is why I called it a grade 3 and not a grade 4. So this is the first marker. Yes, Siddhi, your favorite marker.
IDH.
Very good. How I showed you initially how to denote that. Now, how will you say IDH what?
IDH1 R132.
IDH1, you put a P dot R132H. Okay? Because that is the sequence, amino acid sequence. Alright? So this is R132H. IDH1, R132H. Did you understand? We'll come to that a little later. I will chew your brain on that also. Then your other two favorite markers. Let me tell you what they are.
Left side is ATRX and right side T53.
What does ATRX show?
Negative for the cells, malignant cells.
Negative for the cells. So we'll say that it is ATRX loss of expression. How can you explain that? When there is ATRX mutation, there is no expression. How can we explain? See, ATRX is a gene that is in the brain tumors, mutated in the gliomas in two groups of gliomas. One is IDH mutant astrocytoma and the other one is histone mutated pediatric type of gliomas. ATRX gets mutated. Okay. Now, ATRX was initially identified in other tumors. It was identified in neuroendocrine tumors and all. Then it came to the brain. Now, among these, the type of ATRX mutation is known as, it is a truncating mutation. Okay. Because it is a truncating mutation, the protein is not expressed. So, IHC is a good way to demonstrate this mutation because protein doesn't get expressed. So, none of the tumor cells are positive for ATRX. So, that will tell you that ATRX is mutated. But you must see that there are all these internal controls. The blood vessel, you see this blood vessel? Endothelial cells are positive. Some overrun native glial cells. There will be some native glial cells in the brain, isn't it? They will be positive. Neurons will be positive. If you have some tumor infiltrating lymphocytes, they will be positive. So you should look at the internal control. This is very important because it will also tell you that your stain is working. If you see a negative stain, it may be that your stain didn't work at all. So it also tells you that your stain is working, and this is the loss of ATRX expression. Okay, and this is what P53. P53 is strongly positive. It's not an essential criteria because not in all cases P53 positivity will represent TP53 mutation. Okay, you can only when there is a missense mutation, which occurs in most of the cases, you see P53 positivity. But if there are other types of like types of glycide mutation or truncating mutation or even the deletions, then P53 will be negative by IHC. That doesn't rule out TP53 mutation. So P53 is only a supportive to ATRX. So we use P53 alongside with ATRX, which is very useful for a diffuse glioma diagnosis. So now we found GFAP was positive, OLIG2 was positive, ATRX was loss of expression, and P53 is positive. So the diagnosis now will be astrocytoma, IDH mutant. Even now it is NOS because we are not grading it. We have not put a CNS WHO grade. We have to give a CNS WHO grade. It's histologic grade 3. Okay. So we call it as astrocytoma, IDH mutant, NOS. Even now I'm calling it NOS. Okay. But now, before I go to the grading criteria, I'll just tell you the diagnostic criteria for IDH mutant astrocytoma. The essential criteria is that it's a diffusely infiltrating glioma with IDH1 or 2 mutation and loss of nuclear ATRX expression or ATRX mutation or exclusion of 1p19q codeletion. Sometimes this ATRX loss cannot be identified properly, or sometimes it need not be there also due to different type of mutation. In such a situation, you should do 1p19q and show that there is no codeletion. Okay. So, but we don't need to do that in all the cases. ATRX is more than enough, and all others are desirable criteria. We don't use we are using P53, but it's not an essential criteria. Methylation profiling is a desirable criteria. And look at this, astrocytic differentiation by morphology is desirable criteria because some of these IDH mutant astrocytomas may not look like astrocytoma at all. It can look like oligoastrocytoma. Sometimes it can even look like an oligodendroglioma. So that is the reason we call it, we say that astrocytic differentiation by morphology is only desirable and it has to be diagnosed on molecular markers, and surrogate IHC markers are more than enough to make a diagnosis of this. Now IDH is the most common mutation. Initially it was identified in GBM. We don't want to go into that story. Later it is shown to be a driver mutation in all diffuse lower grade gliomas in infants. Astrocytoma, oligodendroglioma, oligoastrocytoma, all those tumors had shown IDH mutation in the lower grades, in grade 2 and grade 3 and some in grade 4. And the two isoforms are IDH1 and IDH2. Both IDH1 and IDH2 can get mutated in these gliomas. And there are several mutations in IDH1, several in IDH2, about 6 in IDH1, 5 in IDH2. Okay. IDH1 mutation is more common than IDH2, and the most common in IDH1 is this R132H mutation. And luckily for us, that can be detected by immunohistochemistry. This is seen in more than 93% of the cases. So in most of the cases, it is R132H, and we can detect that by IHC. And only in those cases where IHC is negative, we have to look at the other mutations. We cannot, there is no other IHC for that. We have to do a DNA sequencing to identify the other mutations. Okay. So the most common mutation is IDH1 R132H. It is known as the canonical mutation. Others are non-canonical. Canonical by IHC, non-canonical by DNA sequencing. Any doubts? Did you understand this about IDH? So there are two isoforms, IDH1, IDH2, both can get mutated. IDH1 is more common. Out of that, R132H is the most common, and that can be detected by IHC. That is more than enough for you. All right?
I think, Vani, we need to explain, because in clinical practice, the ATRX sometimes interpretation is not so great. And what is the threshold of doing the sequencing? In which case, pediatric type, adult type, forget the G34 and all that, known to complicate of ATRX loss. But if ATRX is lost and IDH is coming negative at IHC, when to do?
Yeah, so this is a very good question that Dr. Rakesh is asking. That is, IDH R132H has come negative, but then ATRX has come loss of expression. It may be in an adult tumor, it may be in a pediatric tumor. Now, the two tumors where there is ATRX loss alongside with P53 positivity in the adult will be IDH mutant astrocytoma, maybe grade 2, grade 3, grade 4. And in the pediatric, it will be pediatric type high-grade glioma, which is that H3G34 mutant tumor. That will also show ATRX loss and P53 positive. How to differentiate the two? Very simple. You use one stain, which is OLIC2. OLIC2 is always positive in these adult tumors in the astrocytoma IDH mutant, whereas OLIC2 is always negative in case of pediatric G34 mutant tumor. So if you have IDH negative by IHC, so this is what I wanted to show you. If you have negative by IHC, if it is positive, don't bother to do anything. If this is negative, in the adult, you make sure your OLIC2 is positive, and then go for sequencing to look for any other type of IDH mutation. In the pediatric age group, if OLIC2 is negative, then you know that you don't have to do IDH sequencing. You can go for histone sequencing, or you have IHC surrogate for that. H3G34R and R and V are the, Geeta will be showing it to you, and they're all the IHC surrogates for that. So please remember, OLIC2 plays a very important role. You can differentiate the pediatric type from adult type when ATRX is lost and P53 is positive. Now, this is very important. What WHO has done is that there is not only a why did I call it as NOS and histologic grade 3 and did not give a CNS WHO grade 3 is because there is a molecular criteria apart from histological criteria for grading these astrocytoma IDH mutant. We know that IDH mutant astrocytomas are grade 2, grade 3 and grade 4. Okay. Now, a grade 2 tumor is a very histologically very well differentiated, no anaplastic features, nil or low mitosis, no microvascular proliferation and necrosis histologically. And molecularly, it will not show the CDKN2AB homozygous deletion. A grade 3 tumor is what we saw just now. It is more cellular, it has anaplastic features, it has significant mitosis, but no microvascular proliferation and necrosis, what we saw in our case. And it will not harbor CDKN2AB homozygous deletion. What about a grade 4 tumor? A grade 4 tumor can look just like this. It can have necrosis and or microvascular proliferation. Then you don't need to do any other molecular marker for it. Histology more than enough. You have shown that it is IDH mutant by showing IDH1 R132H positivity. But you can see that it is grade 4 because of histology. Alternatively, it may have this histology. It may have grade 2 or grade 3 histology. But it can show CDKN2AB homozygous deletion. Now, even then it becomes a grade 4 tumor. So, this molecular marker is very, very important. When in any grade 2 or grade 3 tumor that harbors a CDKN2AB homozygous deletion, automatically gets upgraded to grade 4. Please remember that. And let us see what has happened. What is the CDKN2AB? Now, CDK2AB is cyclin-dependent kinase inhibitor 2A and 2B. It is a tumor suppressor gene. It's located on chromosome 9p locus 21.3, and it encodes for 3 tumor suppressor proteins. It's called P16, P14, and P15. What they do is that they inactivate CDK4, CDK6, or they prevent P53 degradation. And homozygous deletion will result in uncontrolled activation of several genes that are involved in the cell cycle. They transit from G1 to S1, make the cell very proliferative. And several studies, I've just shown one study or 2 studies over here, but several studies have shown that when there is CDK and 2AB homozygous deletion in IDH mutant lower grade astrocytomas, that was associated with a short overall survival. And that is the reason WHO CNSY has picked up this marker for molecular grading of IDH mutant astrocytomas. But let me tell you that the frequency of the CDK and 2AB homozygous deletion is very low in grade 2 and grade 3. Studies have shown that it ranges between 0 to 12% in grade 2. In our own study from my colleague Dr. Vaishali from AIMS, is that we found not a single case coming positive in grade 2. It ranges between 6 to 20% in grade 3. We have seen it to be lower than 5%. But identifying even that small group of patients who has CDK and 2AB deletion is important, because that will be behaving very aggressively like a grade 4 tumor. So that is why the importance of CDK and 2AB is given to this IDH mutant astrocytomas. And in our case, what happened? How do we detect CDK and 2AB homozygous deletion? By the simple FISH technique. I don't know if you read about FISH technique, or you might even have seen those who are doing breast cancer work, HER2 you might have seen the FISH technique, or you might have read about it. So this is the technique where it is histology based, and then we look at interface nuclei in this technique. And here we use a probe against this gene that is targeting this gene, which is CDK and 2A2B located at 9p21, and that is labeled by this spectrum orange. So it will give an orange color, and the control is on the centromere, that is the CEP9 on the centromere of chromosome 9, and that is labeled by green. So generally each interface nucleus must have 2 green and 2 orange signals. But if it has 2 green and no orange signal at all, not even a single orange signal in more than 40% of the tumor cells, you call it as homozygous deletion. If it has 1 orange signal, you call it as hemizygous deletion. Don't bother about that hemizygous. You must see homozygous deletion. So Sidri, can you tell me whether there is homozygous deletion in this or not, in our case? Anybody else? Yes or no?
No,
No, ma'am.
Very good. There is no homozygous deletion. You can see 2 green, 2 orange in all the nuclei, isn't it? So there is no, because they're overlapping nucleus, it looks like many signals. But there is no homozygous. You are seeing orange signals. You should not see any orange signal to call it as homozygous deletion. So our case does not have evidence of homozygous deletion. And because it does not have homozygous deletion, do we upgrade the tumor or we keep it as 3?
3.
It has 3, ma'am. We keep it as 3. So the diagnosis is, now we give a full diagnosis. We give an integrated diagnosis and an integrated grade. Astrocytoma IDH mutant, CNS WHO grade 3. And this is how we put the layered diagnosis. I won't go into details, but IDH was positive, ATRX was lost, and there was no evidence of CDK and 2AB homozygous deletion. And that is the reason that we have given this as grade 3. Is that clear to you? So we'll go on to the next case.
Ma'am, before proceeding, there are a few questions from the attendee. Should I?
Okay. I'm not able to see this chat
Ma'am,
box.
I'm reading to you.
Yeah.
Dr. Tala, I can just quickly tell. 3 questions basically. One was, is necrosis enough to call it grade 4? Irrespective, there is no cellularity, there is no mitosis, there is no atypia, but no MVP, but only necrosis.
It's definitely enough to call it grade 4 if it is an IDH mutant tumor. But let me tell you, as the grade becomes higher and higher, for sure there will be increased cellularity and for sure there will be mitosis. So we will not. I'm talking about an IDH mutant tumor. So we will not have a situation where it is looking with which has no nuclear atypia and with necrosis, and it is IDH mutant. It can be there in some other tumor like pilocytic and all, but not in an IDH mutant tumor. Yes, we can call it grade 4. That's the answer.
No, the question was an IDH mutant only. Second question is importance of P53. It is just to diagnose between oligo or astro, or does it help in grading also?
P53C, this is only a desirable criteria, but we find it very useful on day-to-day practice. It is only a diagnostic marker, no grading. You cannot do any grading with P53. It is only a diagnostic marker, and it goes hand in hand with ATRX. When there is ATRX loss and P53 positive, then we know it is astro. When there is no ATRX loss and P53 is negative, we know it is oligo. We'll be looking at a case subsequently. So P53 is meant only for diagnosis.
Third question.
Yeah, third one.
Third is about which I'll make a comment later. CDKN2AB. Should we do it in every single grade 2, grade 3 IDH mutant tumor?
This is a very, very, very. I mean, this is the question that almost all the clinicians will ask, because we are also in. We should remember that there will be resource limitation in most of the centers, and whether you have the technical expertise or not. Let me tell you that, in my opinion, for a grade 2 tumor, there is no need to do this CDKN2AB, unless, of course, it has all those Pignati criteria that it is looking on radiology, it is a large tumor crossing the midline, patient is older age, then you may want to do it. Recurrent setting, yes, we will want to do CDKN2AB, but I would suggest CDKN2AB for all grade 3 tumors. Because even if you identify that small subset, a small subset of grade 3 will have it. And a small subset also is very important because they will be upgraded to grade 4.
I must tell you, in the last 2 years or so, because we were all thinking whether we really need to do it because it's not available widely, the cost issue, and generally the feeling was IDH mutant, astrocytoma grade ATRX loss looks like a astrocytoma grade 3 in a young person. They should do very well. The median survival is anywhere between 5 to 10 to even 12 years these days. In astrocytomas and in oligodendrogliomas, of course, if they have 1p19q co-deleted, even if it is grade 3 IDH mutant and ATRX will be retained in that. The median survival now with adjuvant therapy is 17 to 18 years in spite of a grade 3 gliomas. Having said that, we have actually one case currently under treatment, young man of 35. Again, similar tumor like what Dr. Vani showed in case 1, right insular astrocytoma, WHO grade 3, MIP1 was hardly 5 to 6%, received adjuvant therapy very well. Within a year, had a progression and had weakness on the left side, had aphasia also, even if it was on the non-dominant lobe. And we did the CDKN2AB and it came out to be positive. So now we are retreating him. So there is a subset of patients as a clinician, because the whole world is telling them the prognosis is very good, don't worry about it, it's not like a grade 4 IDH mutation is very good. Mind you, IDH is a driver mutation. So we have also now in the last 1 year or so, we are doing every case of CDKN2AB in all grade 3. And I also do sometimes in grade 2 if you suspect a sampling error.
Okay.
That on MRI, it does look like you saw the mismatch sign and you saw the midline shift, there is some enhancement, perfusion is high, there is restriction on the diffusion scan, signifying cellularity. But the sampling is such, maybe it is in the dominant lobe on the left side or an allocon cortex, there is only a small tissue, and the histology cannot say grade more than grade 2. And those also we are now doing CDKN2AB. I think these are relevant and which everyone should know. Dr. Tala, any question more? I may have missed more questions you will have.
I think that last point was very, very important what you said, Rakesh. And
one of the questions is that in the case of, in your experience, in oligodendroglioma, the 1p19q co-deletion is positively 100% of the cases.
I'm not able to hear you. There is some kind of echoing from you, Tala.
Ma'am, am I audible?
Yeah.
There is a comment from Dr. Babita. She said that in oligodendroglioma, whether 1p19q co-deletion is positively 100% of the cases. What is your comment on this?
It's a defining molecular marker. You can't call it oligodendroglioma without 1p19q co-deletion. So it is correlated in 100% cases. Okay, that's why it's a defining molecular marker. And there is no oligo without IDH mutation and without 1p9. They're different tumors. They're not oligodendroglioma, IDH mutant, and 1p19q co-deleted. Okay, several tumors can look like oligodendroglioma. In pediatric age group, we have so many tumors that look like oligo, but they will not have 1p19q co-deletion. They'll have different molecular alterations. So
except diffuse glioneuronal tumor can have
diffuse it's called as diffuse glioneuronal tumor with leptomeningeal tumor, that is DLGNTs. They have isolated 1p deletion. Usually don't have 1p19q. They have isolated 1p.
We had one case where they had co-deletion.
Occasional cases have co-deletion, but they'll have a MAP kinase pathway alteration also. And most often it is a BRAF fusion. You should show both. Apart from 1p19q, 1p or 1p19q, you should have MAP kinase pathway alteration, like a BRAF fusion.
Very rare.
So very rare. They are rare tumors. So that's a diagnostic criteria actually for that.
But the more interesting question, Dr. Vani, is we don't have 1p19q fish. Classic oligomorphology, ATRX retained, IDH mutant. We need fish.
So, I think me and Rakesh have been discussing this for many years now.
Yeah.
See, if it is an adult tumor and it has a very typical histology, a typical radiology, without that T2 flare mismatch, a very diffuse, irregular or indistinct margin, some calcification, cortical thickening, whatever it is, Rakesh knows better than me. So typical radiology, typical histology, adult patient, IDH is mutant, you call it oligodendroglioma, IDH mutant, NOS, whichever grade it is. Just go ahead and treat that patient, okay? There is no need to do 1p19q if there is a resource limitation or you don't have that facility. So that term NOS is provided by WHO for that reason only. We have to put that term NOS, which is just a red flag. It doesn't mean failure from on the part of a pathologist. It doesn't mean that. It means that in your center you did not do that, but you know for sure it is oligodendroglioma IDH mutant, and you're saying that it is NOS, which the clinician can read it. Whoever is reading it will say, okay, this is oligo. I'm going to treat it as oligo, and then we'll see later. But there may be some clinicians who will say, no, I'll send it to another center because I would want to know. And there may be some patients who will want to know. I want to know whether my tumor is 1p19q co-deleted because he or she may feel quite gratified after seeing that it is 1p19q co-deleted.
And ATRX retain also will help a little bit.
ATRX retains. Now there are surrogate molecular markers. I don't want to take you on to those surrogates in this session because I will do it separately on a clinical session with surrogate markers, because now we are trying to put guidelines as to how we can diagnose these tumors when there is resource limitation. So don't worry about that. But the answer to Tala's question is that oligodendroglioma 100% are 1p19q co-deleted along with IDH mutation. Is that clear?
Okay, ma'am. Continue.
I think we can't do 5 cases today. We may be able to do only 3 cases. Sumit, please, you need to stop me at some point of time.
Vani, there are so many audience today, and there will be questions. You decide. Please take it easy, whichever way.
Because I'm going to go till 12 midnight otherwise.
No, I think you take your time. Take your time.
So Akshpita, can we have the next case from you?
Yes, ma'am. We have case.
On your video, please.
We have case 2, 35 years old male, presented with generalized tonic-clonic seizures for a year and with no neurological deficit.
So, what do you make out of this history, ma'am? What do you think?
It's since 1 year.
And compared to previous history, is this different?
Yes, previous history was acute and this one is chronic because it has been 1 year since, and there is no neurological deficit the patient has.
Do you think it's deeply located or it's very superficially located?
Ma'am, it can be deeply located.
You think so? We'll have a look at the scan. Okay, good.
Unlikely, because it is seizure. That means a cortical tumor.
Yeah, we don't have it. That's where there is no neurological deficit. If it was deeply located, the patient would have had some weakness of the left upper limb, right upper limb, whichever, lower limb, upper limb, whichever side the tumor is, the opposite side there would have been. But here I'm telling you that it is only seizures and there is no neurological deficit in this patient. Okay?
Yes.
So now we'll see this and ask Kajari to come in.
Yes, ma'am.
One sec, Kajari.
Ma'am, voice is audible, no, ma'am?
Yeah. Your voice is
Okay.
only problem is I'm not able to move it again.
Just click in the center and use your keyboard.
Yeah.
Okay. So we are looking at again 3 sets of images. So first we go to the most right hand side image, that is the T2 weighted image. Again, if we look at the margins of these tumor, again it is a diffuse tumor. So the margin of the tumor is not well delineated. And if we see the location of the tumor, we see that it is located in the gray matter. So rather the subcortical white matter extending into the gray matter. So it's predominantly a subcortical white matter lesion which is infiltrating into the cortex.
Can you point?
And if it is
Can you
point, yeah. So I have to request the pointer, yeah.
Keep talking and I'll point it out.
Yeah. So what we are again, what we see here is that so this is your T2, right? So what we see here is it's a diffuse tumor. It is present in the subcortical white matter. It is infiltrating into the cortex, and if you compare it with the last case, this is the flare image, we don't see any inversion. So basically whatever is the signal in T2, it remains constant in the flare also. So unlike the previous one which had a T2 flare mismatch, this one does not have a T2 flare mismatch, and I don't have a contrast image of this. So basically what I can comment is it's a diffuse glial tumor which is present in the subcortical white matter, and there is involvement of the overlying cortex. It is present in the left parietal lobe, and it does not have any significant mass effect because we can see there is no midline shift or any perilesional edema which is causing mass effect. So therefore the patient does not, and the location of the tumor is in the region that it is not close to the motor cortex also. So therefore the patient does not have any deficits, but obviously this cortical location is responsible for the patient's features, and unlike the previous one, there is no T2 flare mismatch. So
there was let me tell you that there was no evidence of enhancement post contrast.
Okay.
I don't have that image, but
Yes.
it wasn't there.
Other than that, in this particular case, I would also like to have a look at the diffusion and gradient, because gradient can give an idea whether any subtle calcifications are present in this tumor. And in all probability, looking at the location of the lesion, I would put my diagnosis as an oligodendroglioma.
Grade 2 or grade 3?
I think if there is no enhancement and no heterogeneity, it should be a grade 2 tumor.
Grade 2 tumor. Very good. So we'll move on to the next. Okay. Yes, Akhil, can you describe this slide?
See the closely packed cell with small, round, monotonous nuclei and microcalcifications are there.
Is it closely packed? Are they closely packed?
Ma'am, not
Are they
that,
not
but I can
diffused? Are they not loosely dispersed?
yes, ma'am, in this one they are usually dispersed. And ma'am, there are microcalcifications present.
These are all the microcalcifications.
Yes.
So it's loosely dispersed once again on a glial stroma or not on a glial stroma?
Ma'am,
Do you think it is somewhat diffuse? It is spreading diffusely. Can you make out?
yes, ma'am, on a glial stroma.
On a glial stroma, isn't it?
Yes, microcalcifications. Ma'am, here we can see the
Slightly higher
higher
magnification.
magnification. We can see these monotonous nuclei which are slightly larger than the normal oligodendrocyte, and we can see perinuclear clearing, also known as fried egg appearance, which is a formalin artifact. And we can also see the network of thin-walled branching blood vessels, and ma'am, there is presence of these perivascular aggregates of these tumor cells.
So you think that what would you opine? Let's go to the next and then you can opine. Yeah. Now you describe the cell type. First you said that it was low cellularity, moderate cellularity, or high cellularity.
Ma'am, Ma'am, this is in this slide, ma'am, higher cellularity is there.
Moderate. In all the three, it is moderately cellular. And then you can see that the cells are all dispersed on a glial stroma. They're thin branching vasculature, but no typical chicken wire vasculature. There is calcification. Now describe these cells. Describe the cell type.
Ma'am, the individual cell, there is perinuclear clearing is there. Ma'am, with nucleus show a vesicular chromatin.
It is the round cells with a perfectly round nucleus, salt and pepper chromatin, and a perinuclear halo, very characteristic of a differentiated oligodendrocyte, isn't it?
Yes.
Are you convinced?
Yes, ma'am.
Others are also convinced that they are oligodendrocytes that are proliferating. So these are all the neoplastic oligodendrocytes that are proliferating. Now I'll show you diagnostic IHC markers. Yes, Akshpita.
Ma'am, this is positive for GFAP.
What is positive? As compared to the previous one, is it similar to that or different from that?
Ma'am, mostly similar. Like the background tissue is also.
First, you talk about the cytoplasm of the cell, then the fibrils in the background.
Ma'am, cytoplasm haven't stained that well, but the fibrillar.
Cytoplasm is negative. Look at this.
Staining, yes, ma'am.
It is negative. Tumor cells are negative for GFAP, isn't it? What is positive?
But the background
glial stroma is positive. So you must remember that oligodendrocytes are negative for GFAP. Unless you have some other cell type in oligodendrogliomas. There are cell types called as mini gemistocytes, gliofibrillary oligodendrocytes. They will be positive, but not the cells with the perinuclear halo. They will be negative for GFAP. But the stroma will be strong because it is a glial tumor. It is proliferating on a glial stroma, isn't it?
Yes.
And OLIC2 is positive or negative?
Ma'am, positive.
Diffusely positive. It helps us, isn't it?
Yes.
We like the stain a lot. I showed you why it is very important.
Yes.
And so OLIC2 is positive.
Positive.
And how is the labeling?
Ma'am, a low index, low MID.
Low index as compared to the previous one. Isn't it? So this is very low. So what is your preliminary diagnosis and how do you want to proceed with the case?
Ma'am, preliminary diagnosis is oligodendro.
Very good. But do we want to use the term NOS?
Yes.
We use the term because this is histological diagnosis. Now we don't have any molecular markers. It's a molecularly defined tumor. And now we have only histology, isn't it?
Yes.
So it is diffuse glioma compatible with oligodendroglioma.
Yes.
NOS. Histologic grade is 2 because you said that it is low grade.
Low grade.
So I agree with you on that. What next?
Ma'am, then we'll do IHC markers.
Surrogate IHC molecule.
Yes.
Same markers or different markers? What we saw in Astro.
See, ma'am, here we can do almost same markers.
Almost means what? If you want to.
Ma'am, IDH1 we'll do.
Then?
Ma'am, P53.
Very good, and then?
Differentiate
ATRX.
We'll do all of them. The same markers what we did for astrocytoma IDH mutant.
Yes.
So what is the stain on the left and what is the stain on the right?
Here cytoplasm is
This IDH1 R132H. So IDH1 is a cytoplasmic stain. You can see that the cytoplasm is, even the perinuclear halo, you can see how the cytoplasm has taken. In fact, oligodendrogliomas are more strongly positive for IDH when compared to astrocytomas. They're much more strongly positive. And what happened to ATRX over here?
ma'am, positive. With the clear area.
Retained.
You either say loss of expression or retained ATRX.
Retained.
Retained.
Are you sure? Is it lost or is it retained? Be confident. It doesn't matter if you're wrong.
I'm retained.
Very good. It is retained. Why should you feel so hesitant? It is. You saw how it was when it was lost, isn't
Yes,
it?
ma'am.
It is retained.
Retained.
Excellent. Very good. Very good. Full marks for you. Now I have done this stain.
P53, it is negative.
Negative, isn't it? So we have ATRX is positive. IDH is positive, ATRX is retained, and P53 is negative.
Yes.
So what is the diagnosis?
Now we can say oligodendroglioma.
What?
Moderate
IDH.
IDH mutant, you can say,
yes.
but still you have to say NOS because you need one more marker, isn't it? What was the other marker that you require? I answered Tala's question, no? Somebody had asked.
1P19Q.
So that's why it is oligodendroglioma IDH mutant NOS, and because we are saying NOS, we are giving a histological grade. We are not giving a complete CNS WHO grade.
Yes.
Next, what should we do? Test for? Yes, you're right. You can tell me. For which marker now? We have already done IDH. Which is the next marker? 1P19Q, isn't it? And how do we test for that? Again, we do the fish assay. It is tested by fish assay. Here we use two probes. Okay? One probe is for 1P and one probe is for 19Q. We have one test and a control for 1P. The test for 1P is at 1P36 locus. The control is at 1Q25. And for 19Q, the test is 19Q13 and the control will be 19P13. So we use two probes. In every interface nucleus, you must see two green and two orange. Normally, when it is not co-deleted or not deleted. But here you can see that there is two green and one orange signal for 1P, two green and one orange signal for 19. Some cells have zero orange signal. Now, if you see less than two orange signals for 1P and for 19Q, less than two orange, it may be one orange or nil orange. In more than 40% of the tumor cells, then you call it as co-deleted. So in this case, 1P and 19Q is co-deleted. Did you understand? Don't bother about which probe we use and all. It may be a bit too much for you at this level in your MD. Just remember, we have to do a fish test for 1P19Q assay and demonstrate 1P19Q co-deletion. Did you get that? Did you understand that? So now we have everything with us. This is oligodendroglioma, it is IDH mutant, it is 1P19Q co-deleted, and that is the reason we have given a CNS WHO grade 2. And this is the molecular information. It's always nice to put the diagnosis in a layered format. When you put it in layered format, the clinician who's reading it will know what are the tests the pathologist has done. See, each pathologist will communicate in his or her own way to the clinician. I mean, nobody should teach a pathologist that you do this, you do that. Each person has his or her own style. But if you do this, it's a sort of a synoptic reporting. It will tell the clinician, okay, this is how it is. This is the molecular testing they've done, and that is why they've given this diagnosis. And if we do not have any one of this, we would have given it as a NOS diagnosis. So these are the essential diagnostic criteria for oligodendroglioma. It's a diffusely infiltrating glioma with IDH1 and 2 mutation and 1P19Q co-deletion. So you should have this to make a diagnosis of oligo. All others are desirable. Showing retained nuclear expression of ATRX and P53 negativity, third promoter mutation, all that helps in making the diagnosis. But they're not essential diagnostic criteria as per WHO CNS 5. And we have two grades, grade 2 and grade 3. What we saw now was grade 2. Grade 3 was earlier called as anaplastic oligodendroglioma. We don't call that as anaplastic. We just call it as oligodendroglioma, IDH mutant and 1P19Q co-deleted, a grade 3 tumor. These tumors are more cellular, they have increased mitosis. Most often they'll have microvascular proliferation with or without necrosis. And the necrosis, when you see necrosis, it will be very focal. It won't be the diffuse confluent necrosis that you see in case of glioblastoma. So that is a grade 3 tumor. And there are these cell types in oligodendroglioma. You must remember, when you're looking at oligos, you may see different cell types. One is this mini gemistocyte. See, these are small cells that have gemistocyte-like morphology, but the cytoplasm is much more deeply pink in staining, almost acidophilic staining. You find that it will be very, very dark pink in staining. These are the mini gametophytes. Gliofibrillary oligodendrocytes. Sometimes you can see these large cells. Some are spindle, some are polyhedral. They have this fibrillated cytoplasm that is, deeply pink in staining. So this can also be seen in oligos. And some oligos will have such granular cells. They're not eosinophil granular bodies. Tumor cells itself will have granular cytoplasm. And these cell types are usually seen in a grade 3 tumor. When you see such cell types, then you can almost be certain that you're not looking at a grade 2, but you're looking at a grade 3 tumor. Grade 2 tumors are very indolent. Only the perinuclear halo, low cellularity, diffuse infiltration, perivascular and perineuronal satellitosis. That's what we will see in grade 2. But in grade 3, we see these sort of cell types. Not in all. When you see them, then it is easy for you to make a diagnosis. And the term oligoastrocytoma is deleted. It can be used as an NOS terminology, but if you do molecular testing, oligoastrocytomas either become molecularly defined astros or molecularly defined oligodendrogliomas. So that is why the term is deleted. For example, look at this case. This case has both oligo areas and it has astro areas. Do you agree with me?
Yes.
There are these astro areas and oligo areas. So we just call it oligoastrocytoma NOS because we don't know what it is molecularly. But when we did the molecular IHCs, you can see IDH is positive, ATRX is lost, P53 is positive, and CDKN2AB was not deleted. So the tumor became astrocytoma IDH mutant CNS-WHO grade 2. So with any oligoastrocytoma, if you do molecular markers, it will go into either molecularly defined astro or molecularly defined oligo. So the term oligoastrocytoma is deleted and we use it only as a NOS terminology. Is that clear?
Yes.
Both of you have done very well, Siddhi and Akshmita. You were able to diagnose the cases. I'm really happy. But I do want to know whether others have understood this concept, why we use these stains, and when we call a tumor as NOS, and when we call it as a WHO diagnosis. Okay? Is that clear? If there are any questions, I'll take it. Otherwise, okay, these are some key markers. Let me show you some key markers that we use for adult type diffuse gliomas when the histology is either astro, oligo, oligoastro, grades 2 and 3. These are the markers, important surrogate markers, IDH 1 and 2, ATRX P53, Ki67, 1P19Q, and what is included is CDKN2AB. And here you can see that these are the technical platforms. For IDH1, R132H, for ATRX, for P53 and MIB, you can use the IHC platform. For the other IDH mutations, you use DNA sequencing. For 1P19Q and for CDKN2AB assessment, you use the FISH technique. Did you understand that? So these are the technical platforms and these are the set of molecular markers that you require to make a diagnosis of astro or oligo. And astro grade 2, 3, 4 as well as oligo grade 2 and 3. Did you understand that?
Yes.
So with that, we'll go to the next subject.
One small point I wanted to make.
Yes, please.
Sometimes in tumor boards we realize this. There's a bit of necrosis sometimes, and if it looks like an oligodendroglioma, grade 3, of course. So the point is, oligos will never be grade 4. Oligo has only 2 grades, grade 2 and grade 3. Even if it looks bad, cellularity, atypia, mitosis, even necrosis, you cannot go more than grade 3. That is one. The problem comes sometimes is there is oligoastro looking cells, lot of oligo looking cells, but there is some astrostick and there is some necrosis. What do? So you call it oligoastro NOS, but do the 1P19Q. If the 1P19Q it comes co-deleted, you still call it, we used to call it anaplastic, now we call it oligodendroglioma grade 3. But if it comes non-co-deleted, even if it is IDH mutant, ATRX can be either way, actually those times, then you call it grade 4 astrocytoma IDH.
Absolutely.
So I thought,
absolutely. I think Rakesh is a much better pathologist here. His concept is so good. He's really hit it. There's only one point that I want to make over here, Rakesh, and I didn't tell this to the students, that CDKN2AB also plays a role in oligodendrogliomas. A subset of grade 3 oligodendrogliomas which harbor CDKN2AB have been shown to have a very rapid progression. So, and grade 2s will never have it. It will never have a CDKN2AB homozygous deletion. It's not any criteria that WHO has brought in yet, but it does have a role even in oligodendrogliomas. So, when you're having an oligo which has that necrosis and not behaving very well, radiologically not so good, it is worth doing CDKN2AB even in that case, because if it is showing deletion, then you have a problem with that patient. So that is one of the molecular marker which is not really there in the criteria, so I didn't want to confuse the students. So any other question? Rakesh, is there anything on the chat box? Anything is there?
I think it was okay. There were a lot of appreciation, but that's fine.
So we'll go to the third case. Most probably I'm going to end with the third case. Okay, we'll go to Sauvik. Are you there? Sauvik or Nagma, do you want to take the case? Who's taking the case, Sauvik or Nagma? Why so much hesitation? It's a very simple case. I'm telling you, it's very simple.
Glioblastoma. Yes, wild type.
Souvik, please unmute yourself. Souvik.
Souvik, come on, it's such a simple case. Doctor, our chairperson has already given you the diagnosis. You can just go straight for it. Come on.
60-year-old male presented with sudden onset headache and vomiting for 7 days. There is left-sided hemiparesis for 3 days.
What does that tell you, Souvik? What can you make out of this history?
It is an adult
What
brain.
is your impression? Yes?
It is an adult brain tumor and sudden
Just want
onset
to say adult, or do you want to say older adult?
older adult.
Older patient, isn't it?
Older
60 years.
patient,
The others were all younger, isn't it? So this
Yes,
is a 60-year-old male, and what did he present with?
And sudden onset headache and vomiting. That means there is increased intracranial pressure and it is subacute onset 7 days. And left-sided hemiparesis indicate it has a compressive feature.
Very good.
Compressing with the motor.
Okay, so it is a tumor with a acute presentation in an older individual. So let us look. Kajari must come in now.
Yes, ma'am. Can I get that annotation?
Yeah.
So what we are seeing here, we are seeing only two images. The significance of these two images I'll tell you. So when we are looking at this particular area, this one is the post-contrast T1 weighted image. This is the left side, this is the right side. What we are seeing here is this, we should look at this. This is basically your central sulcus. So posterior to it will be a sensory and anterior to it motor cortex. So what we are seeing is this is a peri-Rolandic lesion. What do you call it? This is the area which would be, if a lesion is present in this particular area, that will lead to your weakness. So what we are seeing is the patient is having weakness on the left side. So it is corresponding with the side of the lesion and the site of the lesion. Again, as compared to the previous lesions where the cortex was predominantly involved, we are seeing that the white matter is the area which is predominantly involved. So it's a predominantly white matter lesion. Second, the third thing is that there is significant perilesional edema. So these finger-like projections which we are seeing in the FLAIR image, these are all which signifies vasogenic edema. So there is significant edema which is also again contributing to the features of raised intracranial tension. As we can see, there is sulcal effacement and there is mild midline shift. So these will lead to raised intracranial pressure. And finally, if you look at the enhancement of this, it's a very heterogeneously enhancing lesion with a large area of central necrosis. So the radiology itself tells you that it's a very higher grade lesion. It's a diffuse astrocytoma definitely, but then there is necrosis and heterogeneous enhancement, which point that it is a glioblastoma.
Isn't it? So diagnosis already very simple now. Sauvik, your job is made very easy, isn't it? So
Yes,
okay,
ma'am.
let me. Oh, okay. I don't know. I'm not able to move this for some reason. Maybe you need.
Stop annotating. I stopped annotating.
stopped annotating, but mine is not really moving.
Could you also try stopping annotation?
No, my right click is not working at all. I'm going to unshare and share again, okay? I'm going to share again.
Maybe.
Okay. Souvik?
Yes, ma'am. High cellular yield of, ma'am, I am explaining the left hand side image.
Very good.
There is high cellular yield of pleomorphic population of cell, where individual cell has a nuclear atypia, and there is also presence of high mitotic count, and there is microvascular proliferation, and along with that there is presence of one Brizard multinucleated giant cell.
So, what type of cells are they? Astro, oligo?
Astrocytes.
Pleomorphic astrocytes, isn't it? You can see
Yes,
different types of cells. They are spindle-shaped cells. They are arranged in closely arranged. Sometimes even have this vesicular pattern, some little microvascular proliferation. Then they suddenly become very pleomorphic. Here they are spindle-shaped. They are large cells, gemistocytic
Gemistocytic
morphology.
cells. Yes, ma'am.
Yeah, binucleate cells, multinucleate cells.
Large
so you
eosinophilic
all types of
cytoplasm.
cells. Yeah, so you call it as pleomorphic astrocytic cells. So now what has happened?
Ma'am, here in the left hand side image, I can appreciate spindle cell which is interconnected by reticulin fibers here in the left hand side, in the right hand side, and on the left hand side, ma'am, I can appreciate astrocytic cells.
These are definitely astrocytic. What is this area?
Ma'am, this is sarcomatoid chain.
Here there is, I understand. Here
In
you can
this
see lot of
slide.
spindling, intervesicular arrangement of the tumor cells. So this looks a little bit
Hello.
like a sarcomatoid pattern. But here you can see that there is some necrosis, lot of myxoid change in the stroma. Can you make out that the stroma has undergone some myxoid degeneration? Lot of myxoid change. Some of these glioblastomas have lot of myxoid change in the stroma. So that is what has happened over here. Okay? And high mitotic activity. There is no doubt about that at all. Now you tell me, what is this? Souvik, you're there or you're not there?
Can you listen, ma'am?
Yes.
Yes. In this section, ma'am,
Tell me what is this and what is this? That's all I want.
this is, ma'am,
What
blood.
changes are they? Are they the changes in the stroma, changes in the tumor cells, changes in the vessels? In
Ma'am,
which
changes in the stroma, ma'am. There is a blood vessels are present, ma'am.
what are those blood vessels called? What are these blood vessels?
Ma'am, these are tumor infiltration around the blood vessels, ma'am. Perivascular tumor cell infiltrations.
No, perivascular is there, but when you have numerous such thin blood vessels that come up, you call it as tumor neovascularization. Okay, they're all new tumor blood vessels that are proliferating, thin-walled blood vessels. You know what happens in glioblastoma is that the tumor cells, they throw out a lot of vascular angiogenic factors, several factors. And what they do is they would induct the endothelial cells from the surrounding brain parenchyma, bring it on into the tumor and form these new tumor blood vessels. But these tumor blood vessels they do not have a proper frame, they do not have a proper basement membrane, only the endothelial cells will be forming that. And because of that, it gives rise to extensive perilesional edema that you saw. Okay. So this is known as tumor neovascularization. Another term that is used is sprouting angiogenesis. We also use the term sprouting angiogenesis. Now, what is this one? Another vascular change.
Ma'am, there is some multinucleated giant cells, ma'am.
What is this? Is this a single multinucleated giant cell? These are the tumor cells.
Glomeruloid bodies.
Glomerulate.
Glomeruloid bodies. What are glomeruloid bodies? Why does it come up like that? More simple terminology. It's called microvascular proliferation. Okay. Now the microvascular proliferation is very florid. Earlier we used to call it as endothelial proliferation. But now we know that it's not just the endothelial cells that proliferate. Along with endothelial cells, we have other cells lining blood vessel. Which are the other cells? Vascular smooth muscle cells, pericytes, all of them will proliferate to give rise to these structures, what you called as glomeruloid bodies. We call it as glomeruloid tufts because they resemble the glomerulus of the kidney. That's why you call it as glomeruloid tufts. So this is the typical microvascular proliferation that you see in which tumor?
Glioblastoma.
Very good. You see it in glioblastoma. This type of glomeruloid proliferation is specific for glioblastoma. You can see that even within this proliferation, mitotic activity can be seen. You see that? They're very florid microvascular proliferation. And then now what has happened?
Necrosis.
Here the large area of necrosis is present.
Very good. This is called confluent necrosis. Along with that, what has happened to this is another vascular change. One was sprouting angiogenesis. Another one was microvascular proliferation. Now the third vascular change. What is that? These are very thick, sclerosed and thrombosed blood vessel. This is thrombosed and recanalized. You see that? There is recanalization happening. So actually even the clinician will tell us while operating that there were huge thrombosed veins on the surface of the tumor. He himself will tell us that. So this is another vascular change, and because of this you see this confluent necrosis. Large areas of necrosis which we call as confluent necrosis. So we saw one sprouting angiogenesis, we saw microvascular proliferation, we saw sclerosed thrombosed vessels, and we saw confluent necrosis. So now we will see another type of necrosis.
These are, ma'am, serpentine necrosis. Serpentine pericytic necrosis, where the tumor cell marked the necrosis borders.
Very good, 100%. 100 marks for you. Very good. These are all called as palisading necrosis. See, palisading necrosis can be seen in other high-grade tumors also. Even in grade 3 oligo, sometimes you can see palisading necrosis. But this type of palisading, multiple palisades like this, is very specific to glioblastoma. Okay. So, this is palisading necrosis. Yes, Sauvik, what is your preliminary diagnosis?
Glioblastoma NOS, WHO grade 4.
My goodness, you're too good. Too good. You gave
grade
it
4.
a
Histologic grade
very good. Histologic grade 4, because we are giving an NOS diagnosis. Very good. Full marks for you. What do we want to do, Sauvik?
IC markers.
Marker or markers?
Markers.
ATRX, we can do ATRX. And IDH marker we can do. IDH1. And ma'am,
I have done this. Tell me what is this and what does it show and what is its utility?
Ma'am, it is BRF PV600E mutation.
There you're going, man. Really, from where to where you're jumping. You were doing so well till now. Suddenly you went in some other direction. BRF P600.
Negative IDH1.
IDH1, negative IDH1.
Yeah, it is negative for IDH1 R132H, isn't it?
Yes, sir.
What am I going to call this now? What is your diagnosis? What is the molecular test? Let us not go for any prognostic marker, a predictive marker. We will not discuss that. So, this is glioblastoma IDH wild type, CNS WHO grade 4. Now we know that glioblastoma is only IDH wild type. There is no mutant anymore. Mutant became astrocytoma IDH mutant grade 4. Okay, CNS WHO grade 4. So glioblastoma is only IDH wild type. We are calling this as IDH wild type with IHC itself because the patient is age 60 years and IHC is negative. The reason is, as per WHO recommendation, any patient equal to or above the age of 55 years, if IHC is negative for IDH1 R132H, It's more than sufficient to call it as glioblastoma IDH wild type. Okay? Because the frequency of the other mutations after the age of 55 years is almost nil. You don't get the other mutations. But if patient is below 55 years and IDH1R132H is negative by IHC, then sequencing for other IDH mutations will be required. That you can take a decision based on ATRX status, P53 status, all that you can decide and then do a IDH sequencing. But if the patient is up, this patient was 60 years old and IDH1R132H was negative. So conveniently we can call it as glioblastoma IDH wild type CNS WHO grade. Is that clear? Did you understand that?
Yes.
So in the previous classification, we had these are the two types of glioblastoma, IDH wild type and we had the IDH mutant. And at that time, the essential criteria was that it's a high-grade astrocytic glioma with nuclear atypia, cellular pleomorphism, and increased mitosis with microvascular proliferation and or necrosis. That was the definition for glioblastoma at that time in 2016 classification. And that could be mutant or wild type. But now we know that it is only wild type. There is no mutant at all. So the essential criteria is that it's an IDH or a H3. Histone also will be wild type with one or more of these histological and genetic parameters. So we don't have to have only if we have these one of these two features or both these two looking like this, then sure shot it is glioblastoma. But it may look like a grade 2 or grade 3 tumor histologically. If it harbors any of these molecular alterations, note it down for those who don't know. EGFR amplification, third promoter mutation, gain of chromosome 7 and loss of chromosome 10 copy numbers. If any one of them or more are positive, even then it is a glioblastoma, IDH wild type. So IDH wild type glioblastoma, there are 5 criteria. You can have one or more of any of these criteria. Okay? And the first two criteria are generally referred to as histological glioblastoma and the latter two are referred to as molecular glioblastoma. But both are glioblastoma IDH wild type and they have the same behavior. So just like astrocytoma IDH mutant where you had the CDKN2AB playing such an important role for grading, here also you have these 3 molecular markers which are very important for grading. Okay? Did you understand that? So it is not just histology, but I did not show a case like this, but I'm just telling you that even if it shows grade 2, grade 3 morphology, but it has these features, any one of these features, then you can call it as a glioblastoma IDH wild type. So I won't go into the prognostic marker, but I will just go into the subtypes of glioblastoma. Now, Sauvik, you will be very interested. There are 3 subtypes of glioblastoma. Earlier, we used to call it as variants, histological variants, but now they are called as subtypes. One is giant cell glioblastoma, other is gliosarcoma, and epithelioid glioblastoma. They are the subtypes. Giant cell glioblastoma, these are usually more superficially located tumors, and they're little more circumscribed tumors. And histologically, they have this bizarre multinucleated tumor giant cells. Monstrocellular, that's what we call it, okay? They're GFAP positive, but they will be diffusely, strongly P53 positive. They're IDH wild type tumors. That is giant cell glioblastoma. Gliosarcoma, this is a biphasic tumor. It has sarcomatous element, it has glial element. Sarcomatous elements will be highlighted by this reticulin stain because sarcoma are mesenchymal elements where the stroma will be mesenchymal, and that is why reticulin will be positive and glial element will be negative for reticulin. Reticulin is a very important stain in CNS tumors. You use it for many, for PXA, for gliosarcomas, for medulloblastomas. It's very, very important stain. Simple stain, but it helps a lot. Then we use diagnostic markers, GFAP. If you use GFAP, it is positive in the glial elements, but not the sarcomatous elements. Then you use a mesenchymal marker that is vimentin. But mind you that vimentin is also a marker for astrocytes. It's not just for the mesenchymal cells. So vimentin will be positive for both of them. And there will be evidence of malignant features both in the glial as well as in the mesenchymal element. So that is a gliosarcoma. And the third one is epithelioid glioblastoma. This occurs in younger individuals, older children or young adults. Very aggressive tumor. It has these cells which have this epithelioid or rhabdoid morphology. Very, round to polyhedral cells, abundant eosinophilic glassy cytoplasm, binucleate or uninucleate, nucleus pushed to periphery, high mitosis, and angiocentric pattern of arrangement. All these are very typical. Microvascular proliferation and or necrosis will be there. And these are the tumors, they're IDH wild type. These are the tumors that, not all, about 50% of them will harbor this BRAF V600E mutation. Okay, you were saying no, BRAF V600E? This is one of the tumors that harbors the BRAF V600E mutation, and they're variably GFAP positive. So did you understand that? Very good. You've done very well, Souvik. I'm very happy that you were able to explain. But I just want you to remember that glioblastoma. See, glioblastoma earlier used to be called as glioblastoma multiforme. That multiforme word has been removed, but it's a very good word, multiforme. It's such a variegated tumor, clinically, radiologically, histologically. You'll see so much of variegation. You'll see that the cells will be spindle-shaped, pleomorphic, giant cells, all types sarcomatous, primitive neuroectodermal, all types of cells will be there in glioblastoma. But they've removed the term multiforme, but they are IDH wild-type tumors. So this table is only to revise what we have learned. So among adult-type diffuse gliomas, there are 3 tumor types. The first one is astrocytoma IDH mutant. These are graded as CNS-WHO grade 2, 3, and 4. That has replaced the previous terminologies, diffuse astrocytoma, anaplastic astrocytoma, IDH mutant glioblastoma, which are no longer used. There is grading within this tumor type. That's why there is grade 2, 3, 4. And we use a combined histological and molecular grading for this tumor. Both histology and molecular will be taken into consideration. And astrocytoma IDH wild type is eliminated. Oligodendroglioma IDH mutant 1p19q co-deleted. These are the mandatory molecular markers to make the diagnosis. It's graded as 2 and 3. The current diagnostic and grading criteria, it remains unchanged. It is the same as in the WHO 2016 classification. For grade 3 tumors, we don't use the term anaplastic oligo. We just say that it's grade 3. And the term oligoastrocytoma is deleted. You can use it as an NOS terminology. And glioblastoma is only IDH wild type. It is CNS-WHO grade 4. Note that all of them are in Arabic numerals. And the term glioblastoma IDH mutant is eliminated. And a combined histological and molecular grading is also introduced to glioblastoma IDH wild type. Apart from histology, you take these 3 molecular markers, EGFR amplification, third promoter mutation, gain of chromosome 7, and loss of chromosome 10 copy number changes into consideration for grading glioblastoma IDH wild. Is that clear? Any questions?
Yes, ma'am.
Any questions on glioblastoma? Did you understand glioblastoma? Histological glioblastoma
Yes, ma'am.
and molecular glioblastoma. Okay. So it's almost 9:10. Sumit, can I show another case or shall we stop?
Of course, you can. Please go ahead.
Really?
There are students, there are still, if you look at the chat box, there are still 210 to 208, delegates here in the room. So they will kill me before they do anything else if I tell you to stop.
So, whom will I ask? Come in.
Maybe you can do these 2 cases a little more quickly. That's all I request. That's
okay, so maybe I'll only describe these cases. We have the next case is a 25-year-old male with complaints of seizures for 3 years. That is long duration. Weakness of the right upper and lower limb. This is also of long duration. There is no features of raised intracranial pressure, no headache or vomiting, and the patient had left-sided hemiparesis. So there is some sort of a right-sided lesion. And Kajari, can you describe this a little bit more quickly? Yeah, I'll approve this. Yeah.
you unmute, please? Please unmute yourself.
Kajari?
Yeah, sorry, ma'am. So what we are seeing is these three sets of images. The central one is the T2, which we look at. So first we are seeing that this is obviously a right-sided lesion, and the location of the lesion is in the right temporal lobe. Now, temporal lobe harbors a very distinct type of tumors, and these present with epilepsies. And therefore, temporal lobe epilepsy is a very common presentation of these tumors. Now, in contradistinction to the previous tumors, where we are seeing that these tumors were having ill-defined margin, if we see at this tumor is having a very well-defined margin. There is no significant periodicial edema, and it has two components. So if we see on T2, this has one component which is a little bit intermediate or a little bit solid one, and there is a cystic component which is more posteriorly located. So what we are seeing again in the flare image, this cystic component is showing completely inversion. So it's following the CSF signal, or it is completely inverting, again signifying that this is a cystic component, and this is the solid component. When we go to the post-contrast study, we are seeing that the solid component is showing intense post-contrast enhancement. So basically, there is a terminology in radiology where we say that it is a tumor which has a cyst with a mural nodule. And this mural nodule is a little bit eccentrically located. It is more anteriorly located, right? So we are seeing what is basically a circumscribed lesion which has a cyst with a mural nodule morphology and no significant periodicial edema, which is present in the right temporal lobe. And therefore, my differentials radiologically will be a circumscribed astrocytoma, whether it's a pilocytic astrocytoma or circumscribed, or it could be a pleomorphic xanthoastrocytoma or a glioneuronal tumor like a ganglioglioma. So these will be my radiological differentials.
Excellent. So cyst with the mural nodule, please remember temporal cortex, and she has very nicely described with 3 differential diagnoses. So
Just one point, Vani.
yes.
You know, the T1 contrast is very well seen in the medial near the sylvian fissure, but if you look at the T2 and the flares, there seems to be at the posterior medial aspect of the enhancing nodule some other abnormality. Is that part of the...
Yes, so basically, sir, this part, there is a second component. So if we look at the flare image, we are seeing this dumbbell kind of configuration, whereas only one part of the tumor is enhancing brilliantly. So the entire solid component is not enhancing, and this is a little bit more intermediate on T2. So there may be a little bit of heterogeneity within the tumor also. So then in that case, again, I would again have to look at a gradient image to see whether there is any calcification present within this tumor. And then I would think more in terms of a PXA and a glioneuron and a ganglioglioma rather than a pilocytic astrocytoma.
Exactly, crossed my mind too. You could also do the flare, the diffusion weighted imaging to see whether there's a restriction. It looks to me a pleomorphic xanthoastrocytoma more than a pilocytic.
I think we have some very brilliant clinicians over here. Very difficult for me to... Okay, let me share this again. So here we go. So Nagma, do you want to quickly describe, or do you want me to describe?
Nagma, try. Nagma.
Try.
Yeah.
Try. So you can see a tumor. See, this has come right up to the surface. Actually, this is the leptomeninges. A tumor has come up. It is very superficially placed. Whatever was removed was superficially placed. And let me tell you that it was quite firm in consistency. And here, what can you make out? Sorry? Okay, don't bother about it. I will describe because we are running short of time. You remember Dr. Kajri said that she felt it was more a circumscribed tumor than a diffuse tumor. Here what you can see is a nice pushing margin. This is the tumor and this is the brain parenchyma. There is some calcification. There is a pushing margin over here. See, in the brain, no brain tumor is encapsulated. Okay, with